I would like to present to you our next speaker, Urosh Vilmanovich, Director Clinical Data Operations at Vilmanovich AG. He's an experienced executive holding a PhD in molecular medicine and has a history of working in the clinical information and data management services industry. Skilled in clinical and medical research, clinical data management, biostatistics, medical writing, EDC systems, data standardization, management, public speaking, and customer service. Uros, the stage is yours.
Thank you. Oh, this works. Thank you very much for having me. My presentation is going to be a little bit raw. I jumped in at the very, very last minute, and I finished it somewhere around 10 o'clock last night and sent it to Zlatko. So I actually haven't even gone through it yet. But we'll wing it. I'm used to it. The title of my talk is Clinical Data Management and Clinical Data Science, a Practical Approach. We've heard a number of talks this morning, starting with the AI and the approach that's been happening in Croatia and on pharmacodynamics and pharmacokinetics. Both have certain aspects. I mean, what we do and what I do is relatively close to both aspects of those talks. However, we do truly practical work in that. So literally, Vilmanovich AG, we're a clinical informatics company, and our company has two branches. One is electronic healthcare records. So we develop electronic healthcare records for Switzerland, and our newest one is currently in beta testing at the largest hospital in Switzerland. And the other aspect of our company, we actually work in clinical trials for sponsors, for pharmaceutical companies, and for medical device manufacturers. In that, we have developed our software for it, so data collection, data management, trial management, reporting and analytics, and we also actually even do the biostatistics and write the final analysis reports for pharmaceutical companies for marketing authorizations to see whether a new potential medicine is going to be allowed to give into humans. So we literally do the practical approaches to data management. In that, the tool that we use is Umnia, that has electronic data capture capabilities, so we can create any type of questionnaire form, extremely complex ones for all sorts of clinical trials. Some of them have 5,000, 10,000 questions that are all validated. Document management, randomization, and trial supply. We can track everything that's happening inside of a clinical trial, where every single vial of a drug is, for example. A management system for sponsors, ePro, where patients can actually answer questionnaires themselves that are delivered on time and at a specific time points through their therapy life cycle. And we also have a system, this is all one integrated system for electronic clinical outcome assessments, where we can actually integrate wearable devices, heart monitors, even smartwatches directly into the system in a very regulated fashion. As this is an extremely regulated industry, every single data point that we take in has to be actually validated. And we're talking about sometimes millions, if not for some trials, 1 billion data points at a time. We also provide this type of work isn't just computerized. There are humans behind it, where you have to plan a clinical trial or a protocol. So we have to know exactly, and we take part in planning the clinical trial. We develop the what's called data capture forms. We manage the data. We take part in pharmacovigilance, so actually tracking adverse events, serious adverse events, standardizing them. We monitor all the data entry and the accuracy of every single data point that is entered inside for a clinical trial. Then at the end, we create the data sets, which inside of a very standardized environment. There's regulations as to what a data set for a clinical trial submission has to look like, and exactly what columns, the metadata, the variable names, everything has to be very standardized. Then we perform the statistics and the final statistical analysis report. A little bit of clinical data management, because this is a data science conference. Data management is it's not a new field, and within clinical trials, data management is responsible for managing the life cycle of clinical data. So from collection to delivery, exactly what I just said. But it only focuses on data flows and integrity. The amount of data that's actually now being generated in clinical trials is starting to become inordinate. It used to be relatively simple, maybe 10,000, 20,000, 50,000 data points. Now it's getting into the millions and billions, and it's not possible to do it with standard techniques that have been done even 5 years ago, 10 years ago. So it has actually turned into a different job, which is clinical data science. So this requires domain expertise within the healthcare field, technologies, such as I explained, data analytics, good clinical data management practices, and decision-making through the entire life cycle of the clinical research. A clinical trial project can last years. We're doing one right now that's in its 4th year, for example. And it's going to last another 2. And so also protocol designs, the experiment, because the clinical trial is it's a grandiose medical experiment. Extremely regulated, grandiose experiment, which has everything is planned out in front of it at the beginning. So extremely complex protocols, designs that even predict changes within them in a patient-centric, data-driven, and risk-based approach. So we have to be able to identify risks along the way, ideally in real time, ensuring subject protection and the reliability and the credibility of trial results. Reliability and credibility is key here. The most important part is patient safety. The second most important part is reliability and credibility. Because if the data is not reliable and credible, the trial results will not be reliable and credible, and history has shown that that has happened numerous times. So, the flow of clinical research informatics is planning and designing, setting up of a study, both digitally and as far as human resources are, giving controlled access, enrolling patients, collecting data, monitoring data, closing the study off and closing the database in a very controlled format, analyzing it and decommissioning it. Ideally, we can have lessons learned at the end where you can reuse certain elements of the clinical study for future clinical trials. So, planning and designing, what does that mean for the study protocol? So, what we do in the industry, what is done, when the protocol, when we have the protocol and the study, the study in front of us, we need to actually analyze whether the endpoint or the hypothesis of the study can actually be met whatsoever. This is an example from an actual old study. So how an endpoint is defined, it is literally a statistical hypothesis. So this is the average change in CAT score in participants administered Botox compared to placebo at day 28 from day 1. It needs to be defined in a very specific manner. If this sentence is not written right, you can do everything completely correct in the clinical trial, and the clinical trial will fail. You can spend millions, sometimes hundreds of millions of dollars on a clinical trial, and it will fail. The primary endpoint must be defined in the statistical analysis must be defined very well, and the other most important part is the sample size calculation. You need to be able to predict how many participants you need in a clinical trial. This data analytics, even before the trial starts, is absolutely essential because it is unethical not to have a sample size that is insufficient to meet the primary endpoint, because if it is insufficient to meet the primary endpoint, you will have treated human subjects with a therapy that is bound to fail because you do not have enough subjects to actually be able to say something credible about the trial. The other thing is you cannot have too big of a sample size. You can say, oh, well, what's the problem? What's the problem? We'll just put in more patients. No, you cannot and are not allowed to expose more people than necessary to an experimental treatment. One can spend weeks on just getting one number, and a massive amount of research on just getting one simple number. You need 200 patients, you need 50 patients, you need 2,000 patients. And ethics committees look at this extremely detailed to make sure that you do not have too few subjects and you don't have to have too many subjects. We just did one that if we had one less subject enrolled, the study would have failed. One. But it turned out well. So, the other part of the planning and designing is the subject's actual flow through the trial. What it is that they will be doing at the hospital and when. This is extremely important because the endpoints come out of this and they need to be arranged in such a manner that the data is, again, reliable. If you do certain things too infrequently or too frequently, the data will not be reliable. This is especially, for example, with questionnaires. If you give someone a questionnaire every second day for a month, that is a useless piece of information. Why? Because after the third time, they will just give the same answers over and over and over and over again. This requires experience to actually come up with a chart like this accurately for, let's say, 300 patients to go through and have each patient's data be reliable. So how do we do this? Then we create the questionnaires, the forms. We plan out every single question through the entire questionnaire. We make sure that there are dependencies so that no unnecessary questions are ever given to hospital staff to answer. So if the vital signs were not collected, they should never have to answer these questions, and everything is planned ahead of time. Also, there are checks for answers on every single question, where electronic systems will actually say that you are entering a value that does not make sense. It can even cross-check different questions between different visits, whatnot, and we'll give queries if answers to two questions don't make sense. So you can't say somebody's a male and they're pregnant. That does not make sense. And there are systems that is the simplest, let's say, version of this, but that is how one creates reliable data. That is in a very simple sense, but you get the point. Then we actually have to plan every single question and have a standardized way of setting up a question. Down to that the National Library, the National Cancer Institute of America has actually even given codes for interoperability for, let's say, a kilogram is exactly this code, and every single data point has codes in there that are standardized for interoperability that you can share data between systems. So that when you submit it, they look at the number, let's say, this was a child, 52 kilograms, so this number is 52, exactly this code, that means it's kilograms. And that means it's not just kilograms, it's a weight of 52 kilograms. It has multiple meanings behind it. So every single data point has to have this type of standardization. And that is a regulatory requirement to actually have the data this standardized. Create the forms. From back here, you said, I showed this particular, in a protocol, what's planned. That is exactly like that, one-to-one, reflected inside an electronic data capture system. All the questions are exactly transferred one-to-one, and this needs to be approved by a regulatory authority on top of it. The actual questionnaire must be approved by a regulatory authority. We can also have monitors asking questions about every single data point because the data has to be completely clean at the end. We heard about problems with cleaning data in hospital systems earlier. We cannot have this problem. This problem is not allowed in our industry. When we do, I have calls from our biostatisticians they were screaming that they don't know what to do. And then we have problems with regulatory authorities that actually ask questions even about one data point. Not only that, every single data point has to have a history. We need to have an audit trail of every single thing that is ever entered. Who entered it, when they entered it, what they did with it. So we have, actually this is one data point. This is one question where, are there abnormal signs and symptoms except the underlying disease, and specify if there are. The answer was no. On the 15th of December 2023, at 14:18 and 12 seconds, Sylvia Princhenko did a manual data entry that was the initial data entry. For every single data point, this exists. You end up with billions of data points. And this is extremely important because this actually tells the quality of the study. And we do analytics on this, actually. Not the actual data, but the metadata around it. Because we can actually tell what site is better than others, which doctor is better than others, at which hospitals have patients are at more risk to the hospital staff not understanding what they're supposed to be doing. We can actually detect that inside of the system. This is simple monitoring of a clinical trial, how many patients at which hospital have been randomized. How many have been screened, how many have been randomized, how many are withdrawn, how many have been completed. We can track this in absolute real time. This is questions that people have raised on individual data points. We can detect per site, per query status, whether it's resolved, whether it's active, so it's been answered but hasn't been resolved yet, or it's a new query, and we can go down to the patient level and actually somebody can visually screen what is actually happening at the level of each individual patient. On top of that, we can actually even detect site performance. So these are protocol deviations, and protocol deviations are defined as something that has happened or something that somebody has done that is not according to plan. And every single one of these deviations must be reported to a regulatory authority. So what is important, especially to sponsors, especially pharmaceutical companies or medical device manufacturers who are funding these trials, which are extremely expensive, they want to know how well sites are doing. Are they doing everything according to protocol? What are the risks associated with using that site and using that patient and using those doctors? So here we have the number of protocol deviations per participant by country. This is specifically Bulgaria, Hungary, and Romania. So every second patient in Bulgaria approximately has a protocol deviation. But there are 20 actual hospitals participating in this clinical trial. They're not all the same. And we can actually detect and in real time present to the sponsor. They can click a button at any single point in time and get a real-time report. The system goes in, grabs the metadata, performs a statistical analysis, and graphs this for them in real time. Am I running out of time? I'm not yet. Okay. Sorry, I didn't even go through the talk one before last night before I did this. What we can also do, which is an important measure of quality, this is actually called risk-based quality monitoring. So we're detecting risks, actually, in quality. It is known that the more time has elapsed between when a participant in a clinical trial has had a study visit and how much later the site staff actually entered the data into the system, that the data is of lower quality and that the site is doing a poorer job. So what we actually detect for them is, for each site, how many days after the participant's visit they're entering the data. Now, this is also real-time analytics. So I mean, this is analytics not of the data, but of the metadata, of the data about the data. So we have the overall raw mean. So this is approximately, 16 days after? But the weighted mean, because each site has not recruited the same number of patients, so the system automatically calculates an average of a deviation from the mean, and does a weighted mean to see what the actual average is, and then you can see which are the fastest and which are the slowest. We can also actually even do comparison across carts, a comparison across sites with a Z-course of statistical analysis, and the sponsor has this immediately at their fingertips at all times. And they can actually make real-time decisions for their clinical trial, which could save them potentially millions by intervening, training sites again, and like. So these are just a couple of examples. There's much, much more behind this, but now, so we've All the patient data have been, all the patients have gone through their clinical trial, the hospitals have done what they need to do, the doctors have done what they need to do, all the data is entered. We have all the data, we have all the metadata. And then what now? It's time to actually finalize. So there are 4 steps for the data and for the data and its analytics to actually to be used. Because the entire point of conducting a clinical trial or for a medical device, a clinical investigation, is to actually have the data and data analysis for a sponsor to submit a report to the regulatory authority for marketing authorization, where EMEA, the FDA, Alliance here will say you are allowed to market this therapy, put it on the market, or you're not. So we need to lock the database. All the data, every single the data has to be source data verified. So somebody has to go and check that actually every single data point has been accurately entered, or taken to it by risk the most important data points. There has to be medical review of the important data points. So a doctor actually has to take a look at every single data point, make sure it makes sense. Medical terminology, so for adverse events, for common medications, for medical history, has to be coded. There are standardized coding dictionaries, which actually you can take a drug like ibuprofen, and it has a code for the actual active substance. So ibuprofen here, ibuprofen in the United States, ibuprofen in China is one number. So you can compare actually across patients and across clinical trials. There has to be a data management review. The principal investigator at the site must sign off, and then we can lock it. We can export then the data, create what are called standard data tabulation model data sets, which are regulated data sets with a specified structure. And these are specifically used by the FDA, they're used by the Japanese Medical Authority, and they're used in China. An analysis model data set, because this is the data as was entered, the analysis model data set, because you don't necessarily actually analyze what's been entered, but you can actually impute data and create new data in a standardized fashion, and you have to explain it. And then you have to have a method of actually submitting the data, which is actually creating XML files that can be uploaded to, let's say, the FDA, where all of this is, that contain all of this. Then statisticians have to program the statistical analyses, and this is not done in a manner that, let's say, we learned in school, but they actually create statistical programs to do this, where they analyze the entire data set all at once. These are statistical programs that run sometimes in the tens of thousands of lines of programming code, specifically focusing on the primary and secondary endpoints of the study. The primary endpoint studies with that which has been trying to prove, and safety, always safety. At the end, you have to write the statistical analysis report, which goes into a clinical study report, the final and the regulatory submission to say whether to give somebody a chance as the regulatory authorities to say whether this drug actually, or medical device, works or does not work and is safe. Most important, and is safe. I hope I didn't lose you all. I tried to make it as simple as I could, but to give you an idea in clinical research and in medical research, when you're doing something that is so regulated by the government, how the data flow has to take place, and what we actually do in the industry on a functional level to, what should I call it, to actually carry that out in a practical way. Thank you. Any questions?
If I'm not mistaken, I think I've seen that on your questionnaire there is a free-form text that can be entered.
It's
How is this data handled?
it's possible, but we try to avoid it as much. We try to avoid that as much as humanly possible.
So if there is data, how you guys handle that, and how is this used? Because everything else is coded, and I understand that, but what if there is a free-form data, a free-form text in there?
As far as data analysis, as far as the actual analysis is concerned, for free-form text, we only analyze 3 types of free-form text data. And that is terminology for adverse events. So somebody has a headache, and the doctor at the site writes in whatever they want to write in: pain in the head, headache, whatever. I mean, there's a number of ways that it all means the same thing. That's coded, actually. They're experts, expert coders, using a medical dictionary called MEDRA. It's a medical dictionary for regulatory authorities. And it's an organization that has existed for about 20 years now, which have defined, I'm going to lie to you, about 140,000 medical conditions with each of their codes in a number of layers. And so every single one of those is coded, either throughout the trial or at the end of the trial. That also goes for medical history. The other one is for medications, and that's coded using a dictionary called Who Drug. Who Drug was developed by Uppsala in Sweden, by the Uppsala University in Sweden, and it actually has an AI behind it. So it is possible actually to auto-code it via AI. That AI is not perfect, and a human must go through it at the end, but it helps a lot. The only problem is that Who Drug is extremely expensive to use. It costs about $10,000 a year per trial to actually even just have access to it. But it can do it in real time. And the AI can do it in real time. So that happens in the background. The rest of the verbatim entries are usually things that are really not that important, because not every data point is as important as others. So for example, if something's not done, reason not done, and that doesn't go into an analysis data set, for example. Although we try to avoid in every single circumstance any type of free text.
Thank you.
That's it. Thank you very much.
Thank you very much for sharing your thoughts.
Feedback.
For sharing your thoughts and experiences with us. This is a certificate of appreciation for your engagement in DSC Europe 23. Thank you very much, Uros.
Thank you very much. It was great to be here.
Thank you.